And just as a quick reminder, we are going to be posting this. If there's something you want to go back to, then we are going to be posting this on the GBATnet YouTube, as well as a PDF version on the GBATnet website. So if you want to go back, then that is absolutely possible to do that. So I'm very, very excited to be introducing our speaker this morning. I first got to meet Manny in Houston, Texas over this past year at the GBATnet meeting, and he's an absolutely fantastic researcher, fantastic person just to have around and hang out with. And he is the one that's kind of spearheading some of this incredible technology within the bat world. And up until very, very recently, we haven't been able to age bats. It hasn't been really a possibility other than being able to say this is an adult or this is a juvenile, unless, of course, you had types of information like banding or pit tags. But this is a technology that's going to enable us to do aging of bats to a much more accurate level. And he's doing incredible research. So I'm very excited to be introducing Dr. Juan Manuel Vazquez this morning, who also goes by Manny. So whatever you prefer, let us know. And yeah, if you have questions for the end, we will have probably about 10-15 minutes to do that. Otherwise, I will pass it off to Manny to go ahead and get started.
Thank you so much, Dana. So yeah, great introduction. I'm coming to you all live from Berkeley, and today I'm going to be talking to you about aging at a glance, and specifically the why do we care about bat aging, and how can we actually figure out how old bats are. So for me, the central question I've always been really interested in is the fact that everyone ages. Everyone in this audience right now, whether we like it or not, is currently aging. And everything alive kind of undergoes this like regular rhythm and regular like aging. This is a really big problem in society, because as you may be familiar, society is growing older. And so I'm showing you here data from the US, but this actually holds true worldwide. This is census data of different age groups from the 1900s all the way to the projected estimates after 2020. Although this data is pre-pandemic, so keep, grain of salt. And you can kind of appreciate here that, the amount of young that we've been having has been steadily increasing over time. But as the population has been growing, not only do you have more people being born, you have more people kind of in this like middle working age bracket that's been exponentially increasing, as well as therefore the number of people who are post-retirement age and centenarians over here. And this is an issue for society, not just because society is getting larger, but because a lot of different diseases actually increase in prevalence as a function of age. And unfortunately, many of us are familiar with this with older relatives. In my case, I had a large family growing up, 40 grandparents, great uncles and great aunts. And so from a young age, I actually got to see the aging process unfold. You can kind of appreciate here that the prevalence of the population that has disease really does increase. And you have everything from like arthritis, which just impacts quality of life, all the way to increases in heart disease, cancer, and diabetes, which are serious illnesses that greatly impact our ability to manage the health and public health of different worldwide populations. So there's kind of a big interest in the healthcare sphere to try to figure out how we can make sure that people age healthily. And one thing that's kind of helps us is the fact that aging isn't just about humans. Aging happens to all living organisms, dogs, cats, all of like bats. Every animal that we know of kind of undergoes this like, born process and then eventually ages to like this adult state. And then eventually, becomes old. However, it's kind of weird to think about what it means to become old, right? For humans, it's pretty straightforward. You're a baby, and then you're a toddler, and then you're a wily teenager, a hardworking adult, and then eventually you become old. But for different animals, that means different things. So, for example, you're still a toddler when you're 3 years old, but 3 years old is actually the maximum lifespan of like laboratory mice. So in the time that it takes you to basically barely be able to talk, a mouse has lived its entire full life. And similarly, by the time that you've become like, 27-year-old, you've lived as long as the maximum lifespan have ever been recorded in dogs. And by the time that you reach, like middle age and adulthood, you've basically lived as long as the oldest recorded bat in history. And by the time that you're about 100, you're probably one of the oldest people alive, but you're also about the same age as some of the oldest elephants that have ever been recorded. And so you can kind of appreciate here that, even though the aging process is universal, everything ages, the aging process doesn't happen equally across all animals. Some animals age faster than others. This is especially true when you kind of think about bowhead whales and some tortoises and several other species, which live way past the human lifespan at around 200 to 212 years for the bowhead whale. So it's kind of fun that I can put out all these numbers and cause great existential angst to everyone this early in the morning. But the real question is, how do we actually know how long these animals are living? Like, how am I pulling out these numbers? And so, if you think about it, for humans and for some species, the answer is actually quite simple. We have really good estimates from records, and we have really good record keeping. So here is a photo of Jean-Louis Calment, who is a French woman from the 1800s who lived to be about 122 years old. And we actually are pretty confident that she did live to be 122 years old, because we still have birth records from this church in France where she was born, she was baptized, and then where she died. So because of that, we have really robust documentation that this woman lived this long. And in general, having accurate birth and death records is kind of the gold standard for aging research. There's a lot of people who we think may have lived longer, or people who may or may not, have lived less. But the problem is that if, your church that has, or your temple that has like all your birth records like burns, then, you kind of lost like all that information. Similarly, in case of animals, having captive populations provides a parallel gold standard for lifespan. For example, we have this elephant, Vatsala, who is estimated to be about 105 years old, but we know for a fact from her teeth and just the general record keeping that we have of her that she's been alive since for at least 100 years. So that's still spitting distance of 105 years. And similarly, we have these two dogs here, Butski and Kedvis, both of which lived well into their 20s, and they were actually featured in a big study on like the longest, oldest dogs. But now the question is, how do we know how long they live when we don't have a good captive population? And that can happen for a variety of different reasons. So, for example, in the case of elephants, right, it's kind of obvious you can't really keep a bowhead whale, which is about the size of, it's about 100 tons in a zoo. So, in the case of the bowhead whale, we actually know how long it lives because someone harpooned a whale 200 years ago and then saw the same whale with the same whaling records like 200 years later. And also sometimes at this point, when they live so long, you can actually use some very intense chemistry processes to actually see how long they live. Like, for example, the natural change in chemical composition from things that were born when you were first made things that were made when you were first born. In the case of bats, we have this guy here, Statler, who passed away unfortunately a couple years ago. But they lived to the ripe old age of 34, which is a long time to think if you think about it. The problem is that only a small number of bat species can actually be kept successfully in captivity. And in general, these are these large fruitivorous bats like Statler. And it turns out that these are actually a minority of bat species. Not only that, they're also not the longest lived bat species that we have on record. As I mentioned, there's a bat that we have caught 42 years later. And it's important for me to clarify that it's a minimum maximum lifespan. So if the way that we understand bats right now and how long they live is based on capture and recapture, which means you follow this kind of simple process. First, you catch the bats. And so when I say you catch the bat, I mean you literally catch the bats. And it can be at this point already like a juvenile, or it can be an adult. And if it's an adult, you actually have no way of knowing how old exactly it is because there's not a lot of morphological traits that actually tell you, yeah, this bat is exactly like 32 years old. And then once you catch the bat, you have to mark the bats, and that can be like a variety of different ways. For example, you can band the bats where you put a little aluminum band on its forearm, or on its leg, or on its ear. or you can use the same kind of chips that you use for your pets, but much smaller to actually then place under the skin of bats. And then you can kind of sample or like sense the bats using these pit tag readers, just like at a vet's office. And both of these have the problem that once you release the bats, you now have to hope that nothing happens to that mark. And the problem right now with the state of the art is banding is actually very bad for the bats in the grand scheme of things. A lot of species have reduced like increased mortality after you handle them and you band them because the band starts rubbing against the wing. They don't like it. They try to chew it off. And the pit tags can fall out actually, or migrate, and then you can, or just generally stop working. And assuming that nothing happens to the mark, you're hoping that nothing happens to the bat that you marked, right? And the truth is that the vast majority of bats die due to either infectious disease or due to predation, or they get attacked by cats. So, if assuming everything goes well, nothing happens to the mark, nothing happens to the bat, and you hope that the marking method doesn't hurt the bats, you have to start coming back every couple of years and hope you catch the exact same bats. And hopefully you can appreciate that if a bat lives at least 47 years, 42 years, you're going to spend your entire career hoping to recapture the same bat. And if not, your students are going to try to recapture the same bats. And this is not a high throughput thing. This is not a process that really scales well to populations that are either in very hard to access areas or population, considering the fact that there's over 1,000 bat species, it doesn't translate well across species. So that bat that I mentioned that was 42 years old was a bat in Europe where they caught it as an adult, marked it, released it. Nothing happened to that bat, and somehow they caught that bat 42 years later and released it again. So that's what I mean by a minimum maximum lifespan. It lives at least 42 years because we have no idea how old that bat was to begin with. And then we caught it 42 years later and released it, which means it's probably still around. But all this begs the question, Manny, this is a lot of work. Like, why in the world do we even care about aging bats? Like, isn't it enough that we can like sing happy birthday to everyone and then call it a day? And the answer is aging affects absolutely everything. Aging is like not only pervasive across a tree of life, it's also pervasive across the levels of life. So let me give you an example. For example, that you can think about aging as a process that affects your stress response. You're a lot better able to handle overwork if you're young, and then when you're old, you really don't want to put up with that. If you are older, you have problems with your immunity, you have diseases that I've shown you that increase with age, your behavior changes. I can keep going on and on. Of specific interest to people in our working group is things like, for example, how age affects disease transmission. And so at this point, everyone is intimately familiar with COVID. So if you think about it, right, you have some baseline level of like disease infectivity. In other words, disease susceptibility when you're adults. So in other words, if you're around kids who are like constantly covered in snot and constantly getting sick, you have a certain propensity to get sick. And that's kind of like, it changes person to person, but at the population level, it's pretty constant, right? But then you have kids whose immune system has never been exposed to diseases before. So they're much more likely to get sick. And everyone here who's a parent or has ever had small children around them knows that this is kind of like one of those universal things of life, like death and taxes. And also old people tend to get a lot more sick as well, as it turns out. So the cool thing is that you can give kids vaccines and therefore kids don't end up suffering from all these crippling diseases because all the kids have wrong with them is that they've never seen the disease before. So if you just simply expose them to like an attenuated version of it, then their immune system learns, yes, this is what chickenpox looks like, or yes, this is what the flu looks like. And it doesn't have to deal with like the really bad stuff. It kind of deals with a practice dummy, and then you get to the same level of immunity that you would have as an adult. But the fun thing is that when you're older, you actually need more vaccines every year because your immune system doesn't remember as well. And what ends up happening is that as you get older, your immunity actually decreases, which means that your susceptibility to disease increases and your infectiousness increases. If you're sick, you are able to pass on a disease, whereas if you're not sick, you are not passing that on, right? well, unless you're asymptomatic. But no matter what, in this case, you can kind of appreciate then that as you get older, all these models of how disease spreads across the population, for example, the models that we use to kind of predict the pandemic, the age is a very important component of these models. And, bats are also infamous because there are some diseases that bats do carry, although it's a whole bunch of other species are also uh kind of guilty about this, but bats are unfairly maligned. But the truth of the matter is that we have no idea how this works in bats. We have no idea what the basic immunity of bats are when they're young. We have no idea what's the basic immunity of bats when they're like an adult. And we absolutely have no idea what happens as they get like older and become kind of geriatric, and how that affects their disease transmission. And this would be really nice to know, because if we had this information, we could actually use this for disease models of things like Nipah virus and Hanta that we can kind of use to then govern how we that we can use to govern how we actually like model human bat interactions, livestock and population management, and all that fun stuff to kind of prevent diseases from spreading. Similarly, everyone who's been to the ophthalmologist should be very familiar with this. Aging affects your eyesight. So even though you might have good eyesight when you're young, as you get older, that kind of goes. And this is kind of cute example, but the truth is that this can get actually very serious. For example, night blindness is something that starts happening to you more and more as you get older, where your eyes are less sensitive to light, and suddenly it becomes very hard to drive at night. And you can think that bats' number one not natural predator is windmills. And it's kind of similar to that night driving analogy. If the bats are flying around and they get older, can they still see the windmills just as nice? Or is this why we have an increase of bat mortality with windmills, right? So that's another thing where actually being able to determine the age of bats would really help with conservation goals, because then it lets us see, yes, this population here has a lot of old bats, so maybe we should wait a couple of years for those old bats to like kind of have their natural lives before we start building more wind farms. Like we can just put it someplace else for now, or we can postpone the project a couple of years. And for me personally, I'm really interested in the relationship between aging and aging-related disease like cancer. And this is kind of simple to think about. When you're born, you're a blank slate. And then as you get older, you start acquiring mutations. At some points in your adulthood, you're going to start acquiring all these mutations that leads to you accumulating precancerous cells. And these precancerous cells are special because they're basically one or two mutations away from becoming a tumor. And so by the time you start getting older, then you're much more likely to get cancer. And what I'm showing you here on the bottom is kind of like the data we have to prove that. So every dot is a different tissue. The y-axis is the incidence rate of cancer normalized per like 100,000 people. And this is data from the US Census for cancer rates from 1977 until 2017. And you can appreciate is that at every age group, right? When you're young, it's kind of flat, your risk of cancer. But once you hit your like early 30s, you start seeing this slow and steady increase in your risk of cancer that kind of correlates based on like, how reproductively active your tissue is. Like, for example, your stomach lining is constantly turning and your stomach is exposed to all the carcinogens that you're eating across your lifetime. So digestive cancers have like the highest change with age. But also lung, because you breathe like polluted air and such, and colon, rectum, same thing as like digestive. So the longer you live, therefore, the more likely it is that you're going to have cancer in your lifetime. But we have no idea how that works for bats. And this is really important because by understanding how other species deal with cancer risk, you can actually understand then how they're avoiding it. And if you understand how they're avoiding it, you can learn a little bit about that to help like humans avoid cancer as well. So this kind of brings us to our major dilemma, right? Aging is important. All animals age, and animals age quite differently. And we can use that to kind of inform like a variety of different things, because aging affects pretty much everything. But aging is hard to find out. Banding is dangerous. It's dangerous for the bats. It's dangerous for people who have to constantly go out year after year. And it's very low throughput because you have to go out year after year to try to sample something that lives as long as you do. And also, you can't really keep most bats in captivity. So the number of bats that you can actually age in captivity is very low. So this is the Billy Mays, there's got to be a better way kind of like moment here. Um, we have biomarkers, which are kind of like a proxy that we can measure, some like a quantitative like thing we can measure, um, that gives us a little bit of insight into traits and disease. So, for example, height, like it's a pretty simple one. You can just measure like someone's height. They can be like 5'6" or they can be 6'5". But if I didn't have access to like, like the person themselves, and I wanted to say, hey, I have access to the locker room and I want to know how tall everyone is at this gym, you can look at their shoes, right? And so based on their shoes, it's a pretty good proxy. It's not perfect for like their heights. Generally speaking, tall people have bigger feet. And so that gives you kind of this marker that you can look at. And even though you can't measure the thing exactly, you can actually measure something that correlates so strongly that it may as well be the thing you're measuring, right? Similarly, when you're sick, right, it's very expensive, at least in the US, to go to a doctor's office, get a million tests, and figure out whether or not you're sick and with what you're sick. But you can measure someone's temperature instead, right? And that's another biomarker for like whether or not you're sick. Similarly, it actually turns out that the biomarker for aging is actually inside of all of us, right? Unfortunately, you can't really rely on things like skin because, like how you look, because we all know those people that look like they're 20s and they're like 50s, and we all know those people who look like they're in their 50s and they're 20. But we all have DNA inside of us, and we're all born with the same DNA that we die with. So the DNA sequence doesn't change that much as we age. Um, However, one thing that DNA is based off these four letters, A, C, G's, and T's, that kind of make up the building blocks for all the DNA in all of our cells. But you might stop to think at some point in your life, if all cells have the same DNA, then why is it that we have a heart and a brain that are doing completely different things, both the same genetic code, right? How does each cell know what it should be when it grows up? And the answer is these CPG sites, on top of a whole bunch of other ways that the cell can regulate what genes are expressed and what genes are not expressed, which then determines like whether or not you're a heart cell, whether or not you're a brain cell. These CPGs are, so in other words, a C next to a G, are very special because the body has a way of marking them. It's called methylation. And so basically, every time you have a C and a G right next to each other, here shown as like a blue C and a yellow G, the body can choose to put this like little marker on it, on the C, and then it kind of tells that cell, hey, this region, very special, don't do anything with it. Actually, I kind of want you to like throw it in a closet and forget it existed. This is a silencing mechanism. So in other words, when cells have CPG and methylated DNA, it kind of tells them, okay, here's something I don't want you, you're a heart cell. This is a brain gene. I methylated it. Please do not express brains genes in hearts. And this is really useful because you're born with all these methylation marks across your like genome, across all of your DNA. And it's at birth, all humans, and actually turns out all animals, have a kind of very well conserved set of methylated CPGs across their genome. And as you age, some of these CPGs are actually lost. If you think about it, right, how many of you guys have like a file cabinet that's been around for a couple of years, and then you suddenly realize, oh, well, I don't know where all this stuff is, right? So like, where is my birth certificate? Where is my passport? Where is like, my mother's maiden name? I have no idea. And similarly, in your body, as you kind of get older and older, you just randomly start losing like a methylation here, methylation there. Similarly, sometimes you just randomly gain methylation. Like for example, if you're a smoker, there's a lot of different CPG sites across your like lung cells and your mouth cells that start getting methylated because of chemicals in the smoke, right? And not just smokers, but people exposed to smoke, and like either through smoking or wildfires, or through chemicals, or just naturally, right? Sometimes you just randomly gain a CPG because your body makes a mistake. But the nice thing is that it turns out that while all the things I'm describing kind of sounds random, they actually have their own pace. And so there's some CPGs that like change very rapidly with age, some CPGs that don't change very much with age, and some CPGs that like, don't have any kind of methylation patterns, but they get methylated randomly with age. And in the whole, once if you look across like, 30,000, 40,000, 800,000 of these in the genome, it ends up being a clock where many of these these markers reliably like lose methylation over time, and many of these markers reliably gain methylation over time. And so you can use a technology like microarrays, for example, to kind of ask the question, okay, cool, this is randomly changing over time. What does it look like when a person is like 10, when a person is 20, when a person is 30, when a person is 70, when a person is 100? And so this is really old technology. The way that this works is you would chop up the DNA into a whole bunch of chunks and then have these like little glass slides that have little probes on them. The DNA can bind to the probes, and it depends on whether or not the DNA is methylated or not. If it's methylated, it actually binds more to this red probe. And so when it binds there, the probe glows red on the glass slide, and you can look at that using a microscope. And if it's not methylated, it binds to a different probe. That probe glows green when DNA binds to it. And then you have a whole bunch of DNA, right? And even like the same sequence, you have multiple copies of it across all of your cells. So when you kind of take a skin sample from someone, you extract the DNA and you put it onto this like microarray, you end up getting this nice little rainbow metric from like red to yellow to green. And that gives you the information on every single sequence that you know already that's present on that like sampling array, whether like what percentage of it is methylated. And this was used to great success back in 2013. Well before then by a lot of other people, but in 2013, a scientist called Steve Horvath kind of came up with this Horvath clock, we call it, which is a set of CPG sites that he found very reliably and very tightly correlate with age of the samples. So every number here is actually like a sample that was like taken. And what you can appreciate here is that it's really throughout the whole range of human lifespan, everything from like 0 to almost 100 to like 90-year-olds, right? And this is the age that's predicted by the CPGs here on the x-axis. So here the correlation is 97%, which, as a biologist brings a tear to your eye because nothing ever correlates that strongly. And, the significance of this, like how likely is this to be a fluke, is very small. That's 1 times 10 to the negative 200, which I'm going to be honest with you, I cannot even fathom enough, like what that means. Like that's a very, very, very small number. So in the grand scheme of things, right, you have this training set, which was developed on humans. And it turns out then that you can take these sites and a subset of them, around like 40,000, are actually conserved across all mammals. And so much more recently in 2023, the same group, like a much larger group of people, including Ake Liu is the first author for this paper, basically showed that if you take that and you use these special statistical models, you can actually come up with a methylation clock where it doesn't matter what type of tissue you had, what animal you took it from, it actually works incredibly well across the board. So like this, in this case, we now have a much larger range from 0 to like, 200 possibly. And because, some mammals live 200 years. And the correlation is about 98.3%. So it's actually better than the original clock. But the nice thing is that it also works really well in species that you didn't train in. So for example, if you take all of these like mammal species, you remove all the humans from the data set, and then you ask the question, okay, this data set has never seen a human before. Can we predict how long a human would live with everyone else? And it turns out the answer is yes, to around like, the correlation is around 95, 96%. So it actually is quite accurate for humans when you don't have humans in the training data. Likewise, if you remove bats and then you try to ask the question for this particular bat, how long does it live? The correlation is 97%. So what this means is, this is a method that actually scales really well, not only like across species, because I don't need to have trained it on the species. If I discover a whole new species of bat, a whole new species of turtle, I don't need to go ahead and sample every single individual at every single lifespan to get a really nice training data set. I actually just need to use the data I already have for other mammals and kind of leverage it in this context in order to find out like, okay, cool. I just got a sample of DNA from this species, and it turns out it knows exactly this old. And so you can see how this is actually going to be, if we can get it to work, really, really useful for for aging any bats. The problem, though, is money. And so maybe you've kind of appreciated, based on like all of the like complex things I've talked about, that going out into the field costs money, right? Because you need to bring a whole team of people into some random place. Getting that bat sample costs money, but the methylation sequencing actually also costs a lot of money. So back in the old days of like the like the aughts, right? When we first developed the human genome, it was really expensive to sequence DNA. And the problem with microarrays is that it's a glass slide, right? So that glass slide is good for one individual. I can't really easily reuse that glass slide for more individuals. And even if I could, I'm still looking at one individual at a time. And so microarrays just do not scale with like increasing number of individuals. Like you actually do, like you're physically limited by like a very expensive little piece of technology. However, there's been a whole bunch of new sequencing technologies that have been developed since the aughts, right? So as time has passed, we kind of have this like exponential decay in the cost of sequencing DNA. And when I say sequencing DNA here, I mean actually finding out the specific A's, C's, G's, and T's every step of the way. For all your sequences of DNA that you kind of like sequence in this sense. And then you can also figure out, by just modifying normal protocols a little bit and adding a couple of other chemicals to them, which of those CPGs are actually methylated. And so that gives us a different way to approach this, where we can go into the field, we can sample a little bit of the bat tissue and extract the DNA from it. And we can generate this methylation sequencing library, which tells us where in the DNA do we have methylation patterns. And we can read that library, right? That library that contains all the DNA sequences in your favorite bats, with sequencing, using like Illumina, using PacBio, using any one of these new companies. And the neat thing about this is that you get information about the original bat, like things like, what genetic variants does it have, how you can use that to figure out how closely related all these bats are. There's a bunch of information you get from this. But for the purposes of our project, the most important one is you get all these like nice little reads from your library that you can map to the genome. So in other words, you can map to our kind of encyclopedia of all the genetic sequences inside of that species and figure out exactly where in the genome do you find these CPGs and how does the methylation pattern look? Does it look like this is always methylated? Does this look like it's partially methylated? Which is the exact same data readout that we have from the microarrays, which means that in theory, we can just start using this. And this can go down to like under $2 a sample, because the machine is very expensive, but a lot of sequencing facilities have these and they exist all over the world. And you can generate these libraries relatively cheaply. And so this is a sunk cost. This can be generated cheaply. So you can actually sequence 300, 500, 3,000 individuals for very little money. So the goals of our group then, now that we have this we have this paradigm, other people have already made this work. People have been doing methylation sequencing for humans, for bats, for mice, for a whole number of species. But very few groups have actually been taking, the Horvath clock and the sequencing clock and trying to merge them together outside of humans and mice. So even though for humans and mice we have a nice roadmap, we need to take this roadmap and apply it to bats. And the first step for that is actually we need new training data. Unfortunately, you don't get a lot of DNA from bats if you don't by just taking a little piece of their skin, which, by the way, if everyone in the audience just kind of puts up their hand and pinches this tiny little strip of skin between your finger and your thumb, this is actually the same membrane as the wing. And if you pinch kind of hard, you can kind of appreciate it doesn't hurt very much. You feel the pressure, and maybe if you use your nails, it starts hurting a little bit, but it's nowhere near as much as, for example, doing that to the tip of your finger. And so this is important for me to clarify, because when we catch the bats, we take a tiny little piece of sample from their wing membrane. The wings actually get torn constantly by flying into cacti, by flying into branches, being attacked by predators. So they heal like that. And that means that, we can do this without stressing out the bats too much, and the bats heal like this. The problem is, like I said, it's a small piece of tissue, so it's not like you're getting all this incredible amounts of DNA. So you have to be very kind of judicious about how you're like choosing to use that. So we need more bats of known age that we can kind of get from captive populations, from people who have ongoing longitudinal studies where they're going to the same place year after year and know the age of some of these bats. Uh, and then we can send that out for sequencing. We can send it out to sequencing both using like the old microarray method and using the newer sequencing like shotgun sequencing methods. Um, and we can use that to actually build a Rosetta Stone. We can use that to kind of bridge the old data and the new data together in these like known bat species and kind of translate all the data that we've generated using microarrays for other species to this new sequencing era. And with that, you can actually make an easy and reliable pipeline, hopefully, for any other scientist to kind of like come here and use. And this last part is actually the real key for our short-term goals. The How Old Is This Bat Working Group in GBATnet really is looking to make this a population scale technology that we can bring anywhere. That's not just limited to, the most wealthy like scientists and the most wealthy of nations. Currently, right now, for give you kind of some context, it costs about $125 a sample if you send your samples for methylation sequencing to find out the age. And that's US dollars. So it's already expensive enough for like a lab. If you have 100 samples, that's like $12,000, almost $13,000. Whereas if you had the ability to do that by sequencing, that would actually be probably closer to like, 500, like $600 US dollars. And if you had like 3,000 samples, it probably scales up to like, roughly $1,000. So like it's really good at scale. And also, more importantly, this is something that more countries have access to, that you can actually then send your samples and generally bring the cost down substantially. And if we can get this to work easily enough, the goal is that, if you do field work and you're not a geneticist or you're not like, an aging specialist, you can just kind of take your samples, give it to someone who is, send this out for sequencing from some company, and then kind of get the information back and just generate an age like that. But even then, that still requires kind of you bringing the sample to lab, extracting the DNA, sending it out for sequencing. So our long term is to actually kind of enable us to keep doing this forever, right? Technology is constantly changing. So we want to build a database of known age bats, and we're collaborating with a lot of other groups under the umbrella of GBATnet to get this database of known age bats across the world for future sample collection. And that'll be helpful because this same database actually will help us organize all these longitudinal studies of bats, where we can actually see how different traits change with age of bats. And here we don't actually need to like go in and sample the same bats from birth all the way to whatever age we want to end at. We can use our sequencing technology to kind of come in at a cross section of the bat's life, maybe come back 3 years later, and then see, okay, we caught all these bats, in year one, and we know how old they are, and we know like all of the information that we like looked at, and then we came back 3-5 years later, got this cross section of bats, and this cross section of bats, they're this old, and they have these like spectra of traits, and then you can use this to kind of simplify your life and generate these like really cool data sets of how things change with age without actually having to be there for the whole lifespan. And of course, it would be really nice if we can get a field portable version of this. So in other words, I don't want to have to go back to the lab. I don't want to have to like give this to a sequencing facility. I want to be able to take my stuff, go out into the field, catch the bats, and within 24 hours know what species that was, know the genetics of that bat, and know the age of that bat. And even though I put this under long-term goals, it's actually kind of funny how life goes, because this is actually the one that that's already been done. And I'm showing you here pictures from a couple of years ago, actually, where you can just strap these new really cool nanopore sequencers to your laptop with the very scientific method of tape, which, by the way, science is held together by tape. I hate to burst your bubble, tape and gumption. And then you can actually start sequencing all this DNA, which you can see here, in your car. So like I went to lab and I like did all this like stuff. I put this over here. I walked my laptop to the car, and I want you to know that, basically 12 hours later, the laptop was in my trunk and I was at a concert. And by the time I got back, I had all of my sequencing data done. So the cool thing is that I'm showing you pictures from, a kind of urban setting, but you can actually do this. And I have done this in the field as well. The problem is it's a really cool technology. We need the building blocks. We need everything here. And with that, that's it. I will take any questions now.
Keep fat thumbing my camera and turning it off and on again. Thank you, Manny, so much. That was incredible. I definitely have a better understanding of how this all works and the different applications that we can use it in. So while you were chatting, we did just get like some comments, like whenever you were asking like where people were coming from and if they had any things that they wanted to ask you about. Darian from Prince Edward Island in Canada. So hello from Canada. If they're looking forward to learning if we can get any more specific with age other than juveniles and adults via epithelial, which I think you covered pretty well, that we are going to be, or we are able to age a lot more accurately rather than just adult versus juvenile.
Yeah, so like actually, I can go a little bit more into that. So, there's always some error with these kinds of things. It's not like you'd be able to like know, oh yes, this new bat species that never has been sequenced before. We know it is exactly like 3 years and 2 months and 1 day, right?
Yeah.
But the fun thing is that for mice, the clock is that accurate. I can actually go and say, yes, this mouse has been alive for like 65 days and 2 hours because that's the amount of data that we have. Um, and the fun fact is that the more samples that we get, that's why the long-term goal of the database of known age bats is good. The more people who use this technology, the more data we have. The more data we have, the better the statistics get. And you kind of bootstrap your way towards a day where we will actually know to the day, maybe someday, how old bats live. But in general, right now, our goal is get a couple of years of accuracy, which is still, way better than juvenile and adults.
Oh yeah. Oh yeah. We did have Aiden from Liverpool in the UK. And I don't think that we quite know the answer to this question just because this technology is still pretty new, which is, do older bats survive better than younger bats with habitat loss? That's a great question.
That is a great question.
It's a great
Yes.
question. And this is exactly what bat researchers are going to be able to start being able to answer. So TBD, Aiden.
Yeah.
TBD. And then Noel from oh, sorry, we have another question. Oh, here we go. So does weight influence the age of bats? I guess again, I think this kind of all goes into questions that we're going to be able to answer.
And does my research show a link between bats' diet and age? So I personally don't study that like research. So actually, I'll go in order because they're kind of linked, right? So we know for a fact that in humans, which are kind of a weird thing to like be studying for like weight gain, right? There is like a kind of detrimental effect to being too fat. So rip me. But it's one of those things where we kind of do assume that there would be an influence of age with bats, but it kind of goes in the opposite direction because bats are foraging, and so the heavier they are, the healthier they are because they're eating more. So my guess is actually that if we see any relationship, it's probably like positive. But does the research show a link between bats' diet and health and age? We don't know the age, but we do know one thing about that, which is there's a couple scientists who are studying the stress response of diets, like some of these nectivorous bats. They have constant like levels of insulin in their blood that would make a diabetic like die on the spot, and yet they're not, right? And they have high blood sugar and all this crazy stuff, which we know is bad for human health, yet somehow they are living at least 20 years. So it seems like even though there should be a link between diet and age, because we do see that for mice, for humans, for dogs, for cats, the bats have kind of evolved mechanisms to decouple those two things, which we could actually use to, like, for example, come up with better treatments for diabetics, so that way they can deal with like all this like high blood sugar stuff and not have to deal with like, the negative consequences.
Yeah. The more and more we learn about bats, the more we figure out that we need to start studying them more because of all of these really unique traits that they have. And not to mention, it's also like, I wonder if there is an age influence on the type of fat that is developed within bats, right? We have white adipose and brown adipose tissues. Brown adipose tissue helping a lot with things like thermoregulation and hibernation. So how is age going to be influencing the deposition of brown versus white adipose tissue? All sorts of different things that we're going to be able to start discovering.
Mhm.
Yeah. Well, this is absolutely fantastic. Do we have any more questions for Dr. Vasquez?
Questions, comments, deep existential concerns.
Well, that's the daily. All right. Well, to not try and take up too much time of everyone's morning, we can go ahead and end it here. And again, if there's things that that Dr. Vasquez covered in his talk that you would like to go back to, please visit the YouTube channel or download the PDF. And yeah, thank you. I guess let's all give a really great thanks to Dr. Vasquez for coming and talking with us today. We had great representation from yet again all across the world to tune in to listen to your talk today. So thank you everyone for being here, and thank you, Manny, for an incredible presentation.
Thank you.
All right.
And thank you everyone for taking time in their mornings.
Have a good day, everybody. Bye.